Nucleotides help create the gene which codes for a protein. Here you can see the nucleotides thymine, guanine, cytosine, and adenine. For each thymine, it has a complementary nucleotide base, adenine, on the coding strand, and for each guanine there is a complementary cytosine, and vice versa. Every nucleotide is in a set of 3 called a codon, which codes for a unique amino acid. However, each amino acid can be coded by more than one codon. mRNA transcription begins with a start codon and then begins creating complementary RNA nucleotide bases, like so. mRNA is almost identical to the coding strand of DNA, except the only difference is that thymine is instead replaced with uracil, as seen here. mRNA transcription ends when a stop codon is coded, which stops mRNA production and sends it away from the RNA polymerase.
mRNA processing, there is an addition of a 5' cap at the 5' end of the mRNA, which contains a guanine nucleotide with additional phosphate groups as a result, forming GTP. A poly-A tail containing adenine nucleotides is then added to the 3' end of the mRNA, making crossing the nuclear envelope controlled as well as providing stability. The addition of the 5' cap and the poly-A tail provide important functions such as safely exporting the mRNA from the nucleus by protecting it against hydrolytic enzymes in the near future. Afterwards, specific proteins run with SNRPs in order to remove the intron by joining the exons that were on the other sides of the intron. Splicing is necessary for the movement of mRNA from the nucleus to the cytoplasm. mRNA then exits the nucleus and into the cytoplasm to begin translation.
A small ribosomal subunit then attaches to the mRNA at the mRNA binding site. The first tRNA arrives at the A site, matches its anticodon to the mRNA's codon, and moves to the P site with an attached amino acid. A large subunit then attaches to complete initiation. The amino acids are attached to the tRNA by aminoacetyl tRNA synthase. The amino acid is held here and with the help of ATP attaches to the tRNA. Back at the ribosome, elongation occurs where another tRNA matches its anticodon to the codon on the mRNA strand in the A site, carrying another amino acid. The amino acid attached to the first tRNA is then attached to the amino acid of the newest tRNA in the A site. The tRNA in the P site then exits the ribosome through the E site. The tRNA in the A site moves to the P site and the process is continued until the polypeptide chain has the exact amino acids to form a protein. tRNA can then go to the aminoacetyl tRNA synthase to be recycled. The polypeptide chain is released and the small and large subunits detach, leaving the mRNA in the cytoplasm. The polypeptide chain can then fold into a protein.